Everyone, my name is Jie Tan. I'm a graduate student from the GEISEL School of Medicine at Dartmouth. So today I'm going to introduce you to ADAGE, which is an unsupervised feature extraction approach that can directly learn biological context from big data. So we hear people say that we're in the age of big data all the time, but really how big and how fast the data volume is growing? Here we did some calculations. So based on the ArrayExpress database, we see that the number of organisms with genome-wide assays available grow every year, and the number of organisms with over 500 genome-wide assays available also grew very rapidly. Apparently, data generation is cheap and it's no longer a limiting factor. The more pressing task is to extract knowledge from those data. So for those 2,000 organisms, only a few of them are very well functionally annotated. We don't want to limit ourselves to only study those type of organisms. We want to fully leverage all the available data. And in that case, we would like to think that unsupervised machine learning become the key. So deep learning has really revolutionized how unsupervised machine learning is done. A bunch of people from Google did this crazy experiment. They just randomly capture image from YouTube videos and they then feed them into a deep learning model that is trained on a computing cluster. Then they ask the question, what can the model learn? In fact, the model can actually learn very useful representations, such as a cat face. So the beauty of this type of approach is that you don't need to label each picture whether there's a cat in it or not. The algorithm totally learned this cat from unlabeled pictures. So I thought this is really fascinating, and this paper came out the year I entered my grad school and really inspired my thesis work. So I start to wonder whether we can apply a similar approach to biological data and learn useful representations there. So now let me introduce you to ADAGE, which stands for analysis using Denoising autoencoder for gene expression. So this is a deep learning-based feature extraction approach. To train an ADAGE model, first we will need to collect a large amount of transcriptomic data and then feed them into an algorithm called Denoising autoencoder. So for people who are not familiar with Denoising autoencoder, this part may look very complicated, but don't worry, I'll break it down. So now let's first look at a basic autoencoder. Just as its name suggests, autoencoder is something that will encode input into features and then decode features to reconstruct input. So this algorithm is totally data-driven, which means that it will try to minimize the difference between the initial input and the reconstructed input. Basically, you can consider it as a dimension reduction approach when the feature dimension is much smaller than the input dimension. Then let's move to denoising autoencoder. So we think only dimension reduction is too easy. We will give the algorithm a hard time. Therefore, we will first add some random noise into the input and then ask the algorithm to try to extract features from this corrupted input and then use those features to reconstruct the initial input. So there is a denoise step going on in the algorithm, and that's why it is called denoising autoencoder. So you may wonder, the data themselves are already very noisy because they're collected from experiments generated by many different labs under many different conditions. Then why would you add extra noise into the data? So it turned out that adding noise can actually force the algorithm to learn useful gene-gene relationships so that they can fill in those blanks brought by the noise. So in the Denoising autoencoder, a feature is also called a node. Therefore, we'll use these two terms interchangeably. Okay, so after we have trained an ADAGE model, the more interesting part is to interpret the biological meaning behind the model. If we're dealing with image data, then after layers of training, as I have said, we can learn useful representations like a human face or a cat face. Then what is the cat phase or cat node for gene expression data? Unlike image data where input are geometrically linked, genes are linked by their transcriptional programs or the biological processes they all participate on their biological state. Therefore, we will expect to learn features representing different biological states. And I will show you in a minute whether this is true. Another very important question to ask is how can we interpret a model? So when we think about neural networks, we usually think about it as a black box, which means that it has very low interpretability. However, for ADAGE, we really care about what's the biological meaning behind each feature. And in this case, we leverage the highway genes to help us define a biological signature for each node. And the highway genes are simply the genes contributing highway to each node. So we demonstrate the effectiveness of ADAGE using a bacteria called Pseudomonas aeruginosa. This is in collaboration with the Hogan lab in our microbiology department. Pseudomonas aeruginosa is the optimistic human pathogen and also the most devastating bacteria in the cystic fibrosis lung. So to build an input to the ADAGE, we collect all the public available gene expression data for this organism. Then this, as expected, ADAGE is able to learn very useful representations representing different biological states. And in this model, we characterize two features. One is a node representing what strain is it, and the other feature is representing what is oxygen abundance in the living environment. In this talk, I'll focus on the second feature. So AR is an important transcriptional factor that regulates the aerobic and anaerobic switch. It is in the off state when oxygen level is high and will be turned on when oxygen level is low. So we found that there is a feature whose highway genes are significantly enriched of AR, and genes known to be regulated by AR. So we got very interested into this feature and found that its activity can very accurately reflect AR activity. So here I'm showing you the heat maps representing node 42's activity in 4 different data sets, and each row in those heat map represents a different sample. So the first 2 data sets both have AR knockout, and we can see that node 42's activity would drop when AR is knocked out. However, if you knock out other transcript factors such as ROXSR or DNR, it will not affect node 42's activity, indicating this is an AR-specific signal. Then on the left side, we have 2 other experiments that have changing oxygen abundance, and this is because AR is also modulated by oxygen level, and we can see that node 42's activity are also correspond to the oxygen abundance. So we also conduct a new experiment to further validate that the effectiveness of node 42, and this experiment is done by Jack. He grew Pseudomonas on the airway epithelial cell, and we found that node 42 still very accurately reflect when when AR activity is high or down, indicating that ADAGE can be robustly applied to new experiments. So then we want to generate to see whether ADAGE can be generalized to RNA-seq data because our training compendium mainly contains microarray data. To do so, we first apply the training distribution matching method developed by Jeff. This will allow normalizing the RNA-seq data to a comparable range with the RNA-seq data. I think I have 5 minutes. Oh, okay. Okay, then I'll quickly go over this. So we also compare with PCA and ICA. Basically, we see a very much better resolution using ADAGE in terms of indicating AR signal. And many people ask me, how can they use ADAGE? So given a data set, we can use ADAGE to identify the pathways that are perturbed under between treatment and control. So in this case, the Pseudomonas are challenged with antibiotic, topramycin, and we found that there are two nodes that are significantly differentially active between the treatment and control. And then we will try to find the biological meaning behind these two features. In this case, we can very easily associate them to existing CAC pathways and found that the biosynthesis of steroid groups and the type 3 secretion pathways are perturbed by the topramycin treatment. So what if we're dealing with features that are not mapped to existing CAC pathway? And in this case, we can still visualize them using gene network. So in this gene network, it only contains the highway genes for a specific feature, and they're connected by their relationship in the ADAGE model. So our collaborators found this is very helpful for them to browse this network and see what gene modules are consistently upregulated or downregulated in their experiments. So for the final part of my presentation, I'd like to just share with you our latest work on ADAGE, which is an ensemble version of ADAGE. And the main message is that we observe substantially improvement in performance in ADAGE. And we now can cover significantly more biological pathways and also represent them in a more precise manner. Okay, so I know you now must feel very excited about ADAGE and want to use it to mind your own business. We are currently actively building a web server for it, and it's going to expand it to other organisms as well. In the same time, if you want to use it, we also have the source code available on the GitHub. Yeah, so at the end of my presentation, I'd like to thank everyone from our lab and also our great collaborator in the Hogan Lab and our funding source from NIH Newcomb Fellowship, Moore Foundation, CF's Foundation, and the generous travel funding from ISCB. Thank you.